Good afternoon, everybody. I'm very pleased to be here, and thank you very much. I just want to thank two people before I get started on my talk, and one is, of course, Dr. C. Krishnan, the co-director, for two things. One is he actually nominated me for the IEEE Distinguished Lecturer, so thank you for that, and also he invited me here, so I'm here because of him. The other person who did a tremendous amount, actually, Imran Sheikh, and he did everything, all the logistics to get me here. So thank you both very much. And I hope that can I have a microphone so I can move about? Okay, so what I like to do this afternoon, this is not a single topic exactly, so there's quite a bit of breadth, but my idea would be not necessarily to say all the details in the work, but to give you some clues and give you enough, hopefully, inspiration to see what the work is, and you might get interested. And the other thing I want to say before I get started here is that I didn't know really very much about iBEST, but I sat through many things today, and it seems like a wonderful thing you're doing, and congratulations for that. And the way Sri introduced me in the last, combining science, engineering, and things, it seems to me maybe I should come here and actually join you all. So this talk is going to be about breast cancer. And there are two types, not two pieces, but two types of work I did. One is detection, that is if somebody has it or not, and the other is prognosis, that is these people have it, but how long are they likely to survive, which is a real question for both the patient and the clinician. So it's the detection work that I did before, and so a couple of slides about Brunel and me. Brunel is a young university, comparatively speaking. It's 50-year-old, 52-year-old. And the name Brunel comes from somebody called Isambard Kingdom Brunel. He was arguably the best British engineer, and he lived 170 years ago. And he was an engineer, so the university was set up to specialize in engineering and technology. Nowadays, of course, it's got other things, but nonetheless, technology and engineering plays a much bigger role in the university. And this is a portrait of Brunel. He lived in the early Victorian era. My research interests are quite varied. The two boxes on the right-hand side, are sort of, you might consider standard, somebody in a department like me. But these two boxes on the left are my comparatively recent, in the last 8 years, I've been working in those two areas. And while, as I was asked by one of the co-directors here last night at dinner, which was a very pleasant occasion, by the way, how are these of different things? But I think for me, there's a bridge to connect them all. And that is physics in the sense of modeling, signal processing, and machine learning. And of course, you have to do different things. But nonetheless, I think that's a major bridge which connects these four boxes together. So I just want to say one thing, is this last one, particularly to the young people. The numbers, you live long and the numbers grow. So numbers are not important. And in any case, numbers are destination. What's more important is the journey, because the joy that you get from doing this kind of thing is actually doing the thing, not having got somewhere, actually. And that's the important thing. So I think the research is not, for me, about numbers. It's about challenges, interest, and we need to learn something from it, and it got to be fun. If it's not fun, we are not going to do very well. So breast cancer, we know what it is, but it's a very common thing. Very common thing meaning it's either you know somebody who has breast cancer, or it's very likely you know somebody who has got breast cancer. It's as common as that. And it doesn't, it's not a national problem, it's an international problem. It's not between rich and poor countries, and it's not between Eastern and Western countries or anything like that. So it's really a big problem in every nation. And it affects half the population of the world. The problem is, because it's such a big thing, is that there aren't really enough radiologists to examine all the mammograms. So even if not, they said, common thing in every country, but even if countries like Canada or UK or Scandinavia, you know, where there are some type of national health service, said we want to scan every woman in the land, even if they did say that, that would cost a lot of money. But simply there aren't enough experts to actually go through, because while it's quite prevalent, it's not 50-50, it's a lot less than that. So the false positive, if you like, a lot more people you look through who are fine, and so we need some help, and that's the help to come from computer-aided diagnosis. And that's why the machine learning and signal processing in this, for me, signal processing includes image processing. But if it's not for you, then image processing is necessary, as well as machine learning. And I won't say very explicitly about many signal processing things, but as you'll realize as I go through, that implicitly is all of my work includes that. These are some details, and I won't go through them, but if you got any questions, I will be happy to answer them. Could you give me a time when I should stop so I can time myself? Okay, okay. So this is a picture of a breast, and as you can see, this is the dark region, but it's still part of the image, so image processing needs to sort this out. You don't want to know about this. This is the pectoral muscle, so it's white. But this bit is where the interesting thing may be there. So what one is doing here, what you have to do is, you see white and not so white regions. And X-rays ultimately just the same as light. It's of course different wavelength, but it's the same electromagnetism that determines light, the visible light properties and X-ray properties. So these are scattering. So when you're seeing a lot of white, it's because there are lots of scattering centers. And typically, if the region is dense, then there will be more scattering. And if the region is not dense, it will be dark. Typically, that's the case. And what you're interested in, what the light things are. And then even when you get light things, then you are interested in things like this. And this is called a mass. Mass means it's a possible problem, but it's not necessarily a problem. So you can regard sort of looking for masses and then deciding, is it malignant or is it not? You can look at it that way, two-stage process if you want to. These are some also images, and this is to right and left. And in this red, of course, is not part of the image taken. It's a radiologist expert marked it, and this is something to look at. And this is one where is some described, but it's benign. So there is a mass here, but it's not a cancerous. You have here, and you got slightly different shapes. This is more round, and this is more ellipsoid, if you like. But they're all benign, and there is no problem here. Just to give you some ideas what the images are. Here is actually malignant. So this is a real problem. So these 6 images, they are not the typical of everything, but just to give you some idea what the problem that needs to be solved, this computer algorithm we need to generate. So you look at 6 images, these are all masses, but these two, A, B, are fine, they're benign, And then these are problems. Now, if you look at this, this is very different from all the rest. So you might say, oh, that it's easy to classify this, which it is. But when you look at, but it's not in a much bigger area, but even given these 6, this is also different. But these 4, apart from the scale, they're very similar. Of course, because scale is just a question of how far away. So people farther away, I see the face much smaller, and people in the front, I see bigger. So the size is not really important, because that's just a question of zooming in or zooming out. There are other characteristics that we need to figure out. So that's the problem. How do you tell this is different from that or that, or and these 2 are similar and these 2 are different? These are easier in that sense. So here is a bit of idea for a general artificial intelligence or machine learning. How do you decide? So we have some data, in this case image data, which may turn into feature data, and that's how we do feature extraction. So we don't put the image directly into the classifier. Once we extract the features, so these are parameters that we think might decide or distinguish between benign and malignant. And then a further processing often needed, which is called feature selection. So just because you think these parameters, these features may be important, that doesn't necessarily mean that they are important. So there is a further procedure to decide of these features, which ones may be more relevant to solve this specific problem. And that's what feature selection is about. And then when we have a selected set of features, which is smaller than these features number, then we put it into a classifier. And then we get some kind of decision or interpretation. In the breast cancer, it's a little bit more complicated, but it's not really much. And so we have an image, which was the data before, so that's the same. But now, human expert might look at it. And if not human expert, then we need a machine expert to figure out the masses. And once we get a mass, then it could be, that's the one that we want to look at, not the whole breast. It's the masses we want to look at. So if you're thinking of convolutional neural network, then that needs to work here. Not here, work here, to be more effective. And then this is the same as it was before. So this is a little tool that we need to make. Now the question is, what sort of features, parameters may be interesting or important? Well, generally the things are used shape features. So whether it's smooth or round or well circumscribed or not, and because that's what the fine, benign ones are, but malignant ones are typically speculated, rough, and blurry boundaries. So it's to do with shapes. So that's one set. Another set is called edge sharpness feature. So how sharp is the edge between the mass and the non-mass areas? So you're really looking at the boundary, nothing inside, not just the boundary. And this is interesting. And then the texture features. And texture features over the whole mass. You're looking at the texture features. And so there are 3 sets of features. It's not 3 features. There are 3 sets of features. 1, 2, 3 sets. And feature selection. Why do you want to do feature selection? I'm sorry if you already do machine learning and you know some of these things, but I'm trying to address a more general audience here. So the problem is, if you think about the machine learning, ultimately machine learning is optimization. It's not linear optimization, it's a non-linear optimization. So if you have many features, if you have 10 features, then you have a feature space which is 10-dimensional, and you're doing the optimization in 10 dimensions. If you have 60 features, then your feature space is 60 dimension, and you're doing a 60-dimensional optimization. It is far harder to do optimization in 60 dimension than in 10 dimension. That doesn't mean it shouldn't be done. I'm just warning the practicalities of it. And so you often get caught up in local, when you have a big feature space, you get local optimum. So if you have a mountain, there could be minimum, but those minimum, many of them would be local minimum. There are other minimums. So you want to avoid that. And because it's multi-dimension, so many, the computationally is extremely expensive. So it's these two ways. It's called curse of dimensionality, which basically reflects that you want to work in a lower dimension, if possible. If high dimension is where you must be, then obviously that's where you must be. Otherwise, you want to get rid of that. So if you want to get rid of dimensions, how do you do it? And that's what feature selection is about, is to decide which features are important. So then you work only that feature space with important features in it and forget the rest of it. And there are many, many different types. And there are methods like sequential forward selection. So you kind of choose a feature that's the best among the 10 features if you have. And then you choose the second best. Then you choose the third best. So it's an iterative mechanism. There are also genetic algorithm, which is not iterative. This is much better. And in fact, there are mathematically, you can almost prove, meaning there is enough demonstration that on a larger set of features, genetic algorithm is always going to do better than anything else that you can think of. Then there are types, I'll tell you about this, is independent criteria. That is, if you think about distribution, probability distribution, then to decide whether it's one type of distribution, like say Gaussian, or whether it's a Poisson, or some other distribution, then you want to see how to make these independence criteria. And there are many, many different ways of, there are many things of this type. Because this is to do with class separability. And in this case, class meaning you're saying one distribution against another. And there are already existing things in this area. And there is the other type where you actually include, not only decide you want the best feature, but just because the feature best does not mean it will give the best classifying result. One simple example is football. I'll give you. Oh, by the way, somebody asked me to announce the result of today's football, and that's Saudi Arabia 0 and Russia 5. But I forgot about that. But anyway, so coming back to here, so if you take, sorry, football, I shouldn't say football, should I? I should say soccer. I'm in North America now. So soccer, right. So if you don't know soccer, I'm sorry. But if you do know soccer, you know, there are 11 positions to take up on the field for one team. So you need 11 people. And there are people like goalkeepers, or I don't know what you call them, but they're kind of goalies, whatever, who protects the goal. If you get the 11 best goalkeepers in the world and put them in a team, that's not going to work, even though they're the best 11 goalkeepers. If you take 11 best forwards in the world and put them in a team, that's not going to do any good at all. So you need, they got specific jobs. So it's not just getting the best 11. They need to work together. They need to have the right expertise, just like you're doing in iBEST. You know, so you need to pick and choose. And that is a lot of discretion in here. But here, of course, we try to make it not discretion, much more quantitative. But that's why this feature selection, this wrapper type, is to do those type of things. So you not only get the best, but you want to make sure that this group of best is going to give you the best classifying result. And that's important. So how about classifiers? There are many types of classifiers. There are some things called linear classifiers. So it basically, if you think about the feature space that I said, and there are, you see some picture in a minute, then you have two groups, benign and malignant. You just want to draw a line. Just draw one line. So straight line we're thinking, not a curved line. And that basically is linear classifier. And so there are things like Fisher's linear discriminant analysis, logistic regression, and things like this. So there are things like that. But of course, you know, if you're not in the right feature space, then it is not linear. Then we need nonlinear stuff. So neural networks is one example. It's not the only one. Neural networks is one example of a nonlinear classifier. So you can draw boundaries between two groups, which does not have to be a straight line. It could be a curve. So that's one type. And another type is, which is kernel-based. And kernel-based, because if you know about SVM, that's fine. And if you don't, I'll tell you about SVM. SVM is essentially a linear classifier, support vector machine. And it is great because if we have a linear classifier, we know exactly how to do the optimization. And indeed, Isaac Newton knew it, and he did it. So we know exactly how to optimize. So SVM has the great property of being able to actually solve. So it's not a nonlinear optimization, it's a linear optimization. But then the problem is that SVM, as I said, linear. So that's no good when there is nonlinear boundaries you have to draw. So what then people did, it's a clever trick. And those particular young people, you should learn about it because you may have to use similar ideas or you have to concoct something. And what they did is they brought the linearity in through something called kernel. So SVM that we use and you use, they're actually nonlinear, but that's not the true, that's SVM with kernel. And it's the nonlinearity is in the kernel. So although now you've got the ability to draw curves, the beauty of it is that the parameter space, not the feature space, the parameter space, still you're doing a linear optimization. So SVM as a classifier will always, may not give you the best result, but it is a very good result it will give you. And if you have any other classifier that performs better, you can always tell that's worth having. And some will do a lot worse. Anyway, so, and these kernel-based there are things like KFDA and many other. You know, there is also radial basis function is used in here. Okay, so I'll tell you, I won't go through this, but I'll tell you the same thing in pictures. So these are, I don't remember how many, but I think it's about 57 masses. So remember, so these are real data, real clinical data. So there are 57 masses, and what these PCAs are, you know, principal component analysis. So we took the features, set of features, and we calculated the principal components from those set of features. And then what's being plotted here are the top one, because you know they are ordered. So this is the top one, feature one from PCA, and feature two. And so, of course, there are other principal components, but in this picture, I want to show you something. When you plot in these two, then there are 57, or there should be 57 points here, the masses. So each one is a mass, unique mass of these. And then all the blue ones are fine, and all the red ones are problems, malignant. And when you look at this, it is not possible on this plane to draw a straight line, and then on one side it would be all blue, and the other side would be all red. So what that's telling us, if we use PCA with these, then we can't have a linear classifier that's going to tell us exactly which is what. And this is the basic problem in any classification, not because it's PCA, but you need to look at the data, you know, what it's telling you. And then you know, because if it is clear, separate, then you don't need to use neural network. If they were separate, because why should you? There's a much simpler thing you can use. So it's important, I suggest, to look at the data. And the other thing is, it's not the data, the original data. It's many, many levels of process data, but still it's important to look at it. But you take the same set of data, and now what we do a transformation of these data. Now we are in a different place, different space. But in this space, we still have the same 57 points, same 57 masses. And when you do this, you actually end up with all the blue ones here and all the red ones there. And now, anybody can draw a line. You can draw this one, you can draw this one, you can draw all kinds of lines. Just a straight line, and you don't need neural network to do that. It's easy. But of course, the trick of the sweat and hard work been actually getting to the space, and that's the challenge. And this has all been published in IEEE SMC, Systems, Man, and Cybernetics. By the way, I should have said, all the detection work, well, all meaning vast majority of detection work I did with a collaborator in Calgary, and Dr. Sri Krishnan knows him very well. So, and you can, this is a publicly available data set, the MCI data set, those who knows about it, but it's publicly available from US, Wisconsin data set, breast cancer. And we can get, things like pretty much 99% of success rate. And that's sort of pretty good. And there are many different things. I don't want to go through the details. But basically what I want to say is that you can do a pretty good job. And these are just some of the papers, not all papers, of course. I just noticed that SMC paper is not even here, the IEEE paper. But these are interesting things. But I think if you go on Google Scholar, if you're interested, you can easily find these papers. So this is the picture that I showed you. It hasn't changed yet, but I want to show you something else. And where I want to go is this business about, this is the new bit, that before we have feature extraction and then we did feature selection. There is a cleverer thing you can do, and we are the first one, I believe, that we actually brought it to the community. And that is, we can generate a feature. By generate, I mean we start with these features and we create a new feature. So we don't select. So it's not like, menu system that you can have, either salad or fish or meat, and you choose one of them. Here, we actually cook it. So we make some combination of things. So, and this is, I'm not going to go too much detail about how we do it, but again, it's all published, you can have a look. But this is a way to get simpler classifier. So this is how normally the machine learning works. Data, features, feature selection, classifier, results. But these two, what we did, feature generation, is actually can be put into one. And sorry about that. Oh, there. So this could be, and something like genetic programming is exactly that. So they're not separate boxes. To you meaning any user, this is the boundary you see. You don't see inside anything. You just see this. So you don't have to think about feature selection. You just give the data and it will do it itself. And GA and GP, GA is different from GP. They're of course very close linked. But GP is a classifier with automatic feature selection as far as you are concerned. And it can be used to generate features. And it's this bit that we actually showed it to the world how to do it. And and this is the bit that if we do that, then what we get here is actually minimum distance classifier. So in 2 dimension, it's actually a cut, it's a threshold. In multi-dimension, again, it's still a cut, but it's a more complicated cut. But that's what actually doing here. And without going through the PCA in detail, because of time, I'll show you here. So we trained some data, and these are again real data. And these are the sample numbers, so they're 200. And you see all this data, so in training, everything is one type, and all this came out, everything of the other type. That's exactly how it is. But it's not a great that you got 100% in training data. But nonetheless, having got that, then you go to test a different set of 200. So these 200 and these 200, they're different set of samples. And when you go there, you see all of these do come out red, except there are 3 mistakes. They come out to be +1. They should be -1. And these 2 should be +1, but they're -1. So we made both false positive and false negative. We made both types of error. But we made 5 errors out of 200. So that's 2.5% error. So that's still pretty good result. And what's more, that now we didn't use a complicated classifier. We just use a threshold. We just use MDC. And these are standard things. Okay, maybe without reading the details. Basically saying these are more linear classifiers, and you end up with 90%, just below 90% accuracy. And then you go to this GP, different types, and you end up with 97.5, as I said, it's 2.5, so it's 97.5. And you actually improve quite a lot from compared to before. And there are some references again here. There is that's the SMC, and there's the medical and biological engineering and computing, and there's one in pattern recognition. There are more papers, but you can find them if you look for them. Oh, this is my, he's not here, but as I said, not all breast cancer paper, but this is my collaborator from Calgary. So, one thing to say is that GP is successful in achieving classification with implicit ability of nonlinear feature selection. But if it's a linear feature selection that's needed, GP can do that as well. But the main thing is that it can do nonlinear ones. So GP is a very useful thing to have. It's computationally expensive, but to set it up, but that's only in the beginning. Once you set it up, then it doesn't cost any more to run it. And it's a highly efficient learning tool by combining different nonlinear functions to transform useful information into one dimension, and that's the feature generation that I said. So instead of having 10 dimension, 60 dimension, whatever, we turn it into a single dimension, and that means you basically cut, and that means it's a threshold. So now I want to tell you about prognosis. Now, so this is a case where you actually have people who have breast cancer. You know that, they know that, but you take samples, but this is using genetic data from, and what we're doing sophisticated clustering. So I want to do first 2 things. I want to tell you what clustering is, a quick introduction. And then I want to tell you how, are we using just standard clustering? What are we doing? And there is something novelty there. I want to tell you about that. And then I'll quickly give you some results. So if you don't know clustering, then what's clustering? And clustering is we are given a set of objects, whatever they may be. So in this case will be of course, a set of genes, because we are looking at genetic data. Each cluster then represents a set of objects that are similar. And different clusters represents different sets of objects where different sets are dissimilar from each other. So this is some way they're similar in a cluster, all the objects, and between objects between 2 clusters are different some way. So I want to give you a demonstration in case you haven't seen it before, the clustering. So by pictures, and we just do the pictures in 2 dimensions, then it's easy to see, and pretend that there are no colors on this. I know there are, but just pretend. So they're just black and white. So we got all these objects, so each dot or squares, whatever you see them, each of them is an object. And our job is to put them into this set of objects, how many clusters are there. Now, your brain is pretty good at it. And you can see 1, 2, 3, 4, 5, 6, 7. 7 clusters. That's what 7 clusters are. And that's why I colored it separately for you to appreciate that. And these are easy. You might argue, you know, these couple here or 3, should they be green or yellow or lime green? You can argue these 2, should they be blue, light blue or red? But generally speaking, they're easy. So these are fairly compact clusters individually. They're convex, they're closing in, and they're separated. They're big gaps, generally speaking. So these are easy thing to do. Then you go to here, and the things that worked last time, the kind of thing that you optimized to get that, they don't work actually here. And the reason is, while there, this is convex, this is actually concave. So same algorithm wouldn't work here. But nonetheless, your brain is happy to accept that this red and this red, despite they being so far apart, are in the same cluster. And yet this red and this blue, being so near, is not in the same cluster. So the Euclidean distance is not going to help you here. It's not going to give you the right decision. So one needs to be doing it in a different way. And this is even a harder problem. So here again, your brain is pretty good in figuring out that all these red ones really belong to a cluster. And all these blue ones also belong to a cluster, and they're different clusters. And so is the green one. But if you think about the blue one and this blue one, who are in the same cluster, and you go across, you actually cut through red cluster, green cluster. So again, Euclidean distance on this plane is not going to help you solving this. So that's the kind of, and there are of course other pictures. That's not the, I just wanted to give you a flavor that the challenges that you face in clustering. So if you are interested in clustering, we published a book by Wiley in 2015, 3 years ago. And it says integrative cluster analysis in bioinformatics, but you can almost delete this. And there's a lot about integrative cluster analysis. So if you're interested, it would be worth looking what's in there. So I want to say, if you're not motivated in clustering, I want to give you a couple of motivation. One is, why to do clustering? Okay, so hypothesis-driven research is what Francis Bacon kind of addressed. And so he basically said, you start with a white sheet of paper and look around, and you write down a hypothesis, you do the experiment, and if the experiment says the hypothesis is likely to be right, you hold on to that belief. If it says no, then you change to a different hypothesis. That's fine. That's very much focused, I would say, and restricted. Now, why focused? Because you already made a hypothesis. I mean, you're only checking that and nothing else. So that's focused. Restricted, also because of the same reason. Now, and to be able to write a hypothesis, you need some background information. So you need a certain basic knowledge in that area to be able to make a hypothesis. Otherwise, you can't make one. And so data-driven research is different. It's more defocused and more open. So you basically don't know what there is, but you're looking for it. And that's what exploration is. So oil exploration is done very much that way. Darwin did not come up with evolution. He didn't, before he went on the voyage of the Beagle, he didn't write down on a piece of paper, I think this is how evolution works. And he went around and he said, look, there's the evidence. No, it didn't work like that. He went on the expedition. He saw many, many things. And somehow, at some point, things began to emerge in his head. And that's what he wrote down. And then he checked those things. And then it all worked out. That's how it happens. Any big paradigm shift does not come from actually evidence first. It comes from somewhere else. General relativity did not come from experiment first. Einstein did not come up with that. Any large paradigm shift, always to do with exploration, whether that's in mind, or in nature, that's different, but it's with exploration. Exploration is what is necessary. So depending on the stage of the field, area that you're working in, some field may be very highly, researched, and you know an awful lot about it. Then you don't really explore. Then you know what the gaps are. You go for those gaps. When something is completely open, you have no clue what the gaps are. You don't even know anything about it. So then you need to explore. And then clustering is the way to explore. And perhaps Darwin was the first person to cluster. He didn't realize that, but we do. So, and then I want to motivate another thing, consensus clustering. Why do we need consensus clustering? You might even say, what's consensus clustering? I'll tell you. So, there isn't one clustering algorithm. There are many. And there are many different types of clustering algorithms. But there are many algorithms. So you give the you take many algorithms and you give the same data to each algorithm, it will give you different clustering results, different sets of results. But you are after one truth, and you got different versions of truth maybe. Or maybe one is right, everything else is wrong, or maybe all of them are wrong. So how do you know which one? So that's what you need to figure out. Or you give the same clustering algorithm, but you get different data sets because they're similar. You do many experiments or something like that. And you end up with different results because it's different data sets. Again, you are after one truth, and you don't get one truth, you get different. And worse than that, you can have the same algorithm and same data set and run many different times. You get different results because many of these have stochasticity inbuilt into these algorithms. And because of stochasticity, every time you run, it will take a different random path. And because of that, it will give you different results. So this plurality of the results is not helpful. So somehow, you have to bring all this plurality of results and make it into one set of clusters. And that's why you need consensus. So somehow you have to bring that. And we are the first one to do that in the world. And what we did is through genes. So we have, all of us have the same set of genes, meaning humans. So there is some fixed points is the set of genes. And let's say we have different sets, microarray data set, different sets of data. And we take one set, one method, we get one partition, sets of cluster. We get another, same data set by another method, we get second partition. And there are L data sets and C methods, we get L times C partitions. So we got all these R partitions, but we only want one. So how do we do that? And how we do it is doing this. And this was published in PLOS One 5 years ago. He's a medic in Oxford. So this is how we do it. Well, it doesn't say exactly how, but schematically it says, so we take all of these partitions and we put it into here, and then we introduced a new thing called binarization technique, and we actually use 6 different ones. And then once we go through that, then we end up with actually one set of results. And it's got many advantages, but I'll probably just go ahead and come back later to tell you about them if necessary. And so it's 5 years ago, it's already been viewed more than 4,000 times. Okay, and we also introduced, no, I'll miss this, sorry about that. So what we did is for breast cancer, I said the prognosis. So we took the data, there are 16 data sets, and we used 3 algorithms. So that means we got 48 sets of results, but we have to create one set of results out of these 48 sets of results. And we've done that. And one is called cluster 1, that's got 504 genes, another one, cluster 2, is got 598 genes. These are different genes from these ones. There is no overlap between them. And what you might say, what are these things? What these things are, each one, data set 1 to data set 16. So each one is a single data set. And This is all cluster 1, and this is cluster 2. So it's the same 598 genes, cluster 2, and these are cluster 1. And the x-axis is time or conditions. What's interesting for biologists is that these genes, they actually go down and they go up. These are expression values, average expression values over these, all these genes, average of 504. This line is average of 598. These are error bars or variation. And these go up and they kind of stay roughly the same, go down a bit. And when you calculate the cross correlation between this set and this set, it's almost -1. This set and this set almost -1. In every pair of correlation of every pair in each data set is almost -1. And they're all less than -0.95. And that was a surprise. And I asked, because this work I did with somebody else, also from Oxford, a world-renowned oncologist, breast cancer oncologist, and he said, biologists don't know about this set. I said, why is that? He said, because these negatively correlated things we never looked for. So it's the hypothesis-driven. So they look for things that positively correlate with breast cancer. They never look for things that negatively correlated with breast cancer. So they missed out the whole set of genes. We did not look for either, positive and negative. We just looked for clusters. It just turns out that they're positively and, this way they're correlated. So this is the power, another reason, extra reason to say, this is the power of exploration, that you find things that you didn't look for. And then what we can, out of these, there's the correlation plot, there's 16 stars, and they're all below -0.85 or something. And these are too much detail, but basically we have, out of these, we got a set of genes which can give much better prognosis than anything that exists at the moment. It's always as good, and oftentimes is better than what exists now. And this has been published here in Molecular Cancer last year, so almost exactly a year ago, actually. And this has got many people in the area interested. So, what's next? Okay, before I go there, I want to tell particularly young people is that, as you can see, these things, they're not IEEE papers. I've got nothing against IEEE, as you know. I'm here probably because of IEEE. So, but what I want to tell particularly young people is that anything that you want to build big, you need different types of people with different expertise, because that's how everything is built. It's like this room. It's not just bricks or glass. You've got chairs, it's got computers, it's got lights. You need different types. And that's how anything big is built. And so that means we need to work together. We need to be interdisciplinary. And while you're doing it, not when you get there, but while you're doing it, I believe we should take the message to the people who may implement it to make the change. Because ultimately, we create tools. It's no good creating a hammer that never gets used. What's the point? It's no good designing a car or making a car and nobody drives it. Why? What do you do that for? So that means engineering is about problem solving. If you solve the problem, then let other people use that solution to solve more problems. That's what we should be achieving. And so I always try to publish where I think would be more engaging to the community. I want them to change. It's hard, but I can try. So closing remarks is that these two are names of this consensus clustering algorithm. They're all published. And I think they provide new paradigms. And not only I think now people, they're the first ones, but now already people are bringing out other algorithms to do these kind of things, which is good, because I'm not saying these are the best, but we actually set a trend and people are already starting to publish things, and that's good. And that's how we should go. If we see something good, we should make it. And in chess, well, I'm not good at anymore, But when I was young in university, I used to play a lot of chess in tournaments. And I remember one of the grandmasters said, if you're playing chess, he said, if you find a good move, don't make it. He said, find a better one. So that's the idea here. Okay, so data-driven methods produce results of interest to practitioners in the field, and so we should take it to that field, not put it in other places where nobody knows about it. Nobody who can make a change knows about it. And I think this is very, very important, that for big data and complex data research, I truly believe signal processing and machine learning are vital to harness the power of interdisciplinary research. And more than that meaning as well as that, we are in a very special and exciting period. There's a whole lot of developments that have happened. I can name them. One of them is, you know about PCA. A PCA is essentially a matrix factorization, SVD, singular value decomposition. So that's a matrix factorization. But when you're talking about breast cancer data, many of the things you spoke about today, they are multidimensional data. Sometimes there's videos, so they're already three-dimensional, and there are many more than that. And so what we need to do is how do we factorize these things? If you think 20 years ago, there was nothing like that existed. But now we have things like non-negative matrix factorization and many, many. So there are many, many tools are available now that they weren't even 20 years ago. There are many devices available now that weren't. So for young people going forward, it's an enormously advantageous time to make big gains. And medical physics or medicine is crying out for it. I can give you, somebody was talking about red blood cells today, I think one of the students in competition. There are many things, even simple things like blood donation. If you look at how people decide, because donation is people go, I give blood now, and then 6 weeks later, I don't know in Canada what the rule is, 6 weeks later or whatever, I might go back and try and give blood. And often happens that my hemoglobin level is not high enough, so I can't give blood. But that costs money both for the people who are taking it, for me, and I might get fed up. I say, well, I don't want to give it anymore. But we give blood, we need blood. So there is a whole process of prediction around it. Who can give blood and who can't. I don't know in Canada, but I'm sure there is. And they call people. But the strange thing is, if you look at this prediction, don't quote me. I hope it's not broadcast. The methods that are used, all the significant developments in prediction that has happened in the last 2 or 3 decades, I would say, remains untouched. These prediction methods don't know about them. So, there is a vast improvement you can make, and right now I'm actually doing it with some medics. But so I think it's a great field, and I think what you're doing here is good. So thank you very much.
Thank you very much, Professor Nandi, for a very inspiring talk and really doing something called explainable AI. In the world of AI, many things are not explainable, but Professor Nandi took the effort to explain on the new aspects of feature generation and so forth. But we'll have time for 2 quick questions, maybe from the audience. Please feel free to use the microphones on the sides.
You took us from the genes from the images, macro, to the genes. So what are the challenges to be able to combine both of them? The genes they have a level of organization that is at the molecular level, and the other is macro level. Are they, how do you take into account the level of organization in these models?
I think that's a good question, and I think about it, and I think 1 or 2 little things maybe in there. I didn't talk about it because the time, but I'll tell you something else since you helped me to go into there, and you all know about it. It's talked about personalized medicine, and as you very well know, that for the same problem for the same medical or health problem. If 10 of us have the same health problem, the same drug does not necessarily work very well for all 10 of us, but certainly not for the whole population. So it's not that, oh, I got X and you got X, so I need drug Y and you also need drug Y. Not necessarily. So there is some personalization that's needed in medicine, and that will come. That will come not in 5 years, but that will come, I should think, in 10 years and beyond. And there's a lot of effort going in, I'm sure, everywhere, but I can tell you at the moment it's going in UK. And I'm looking into that. And that's where I think is actually gene level is better, because gene is more about you. And it's not that the image isn't, but the image is already something started happening. And the prediction, if you think about the prediction, meaning one is to detect and cure, if possible. The other is to predict, so that before. Now, at image level, the prediction is very difficult to do, i.e. when there is no breast cancer, maybe a little thing, and then to say, oh, you need to look at. But gene may be a better way to go. So there are two aspects. One is personalized medicine, and other is prediction, But of course, image is, and these days, gene analysis is so cheap and so quick. Images are much more difficult. Difficult meaning not difficult in the sense that you need to go somewhere, because you can't have it easily. These are X-rays, so you got to be protected, and people who are taking the pictures, they need to be protected. So there is some specialization necessary. So I'm not saying we should give up on images, but I just feel that the gene is quite a good way to go.
Thank you again for a very good presentation. I just want to ask a question about, again, that really neat idea of clustering and how you found the anti-correlation in clusters of gene expression. So my question is more, what would you advise young faculty or young investigators that now, especially through initiatives like these, will have access to so much of that data and will be able to do so much of this exploration, yet if they apply for a grant, or if they, try to get funding for that, it's very difficult. I mean, I've been on many grants panels where the idea of this kind of exploratory research, they call it a fishing expedition, and they kind of, say that it's not hypothesis-driven and therefore it won't be consequential. So from your experience or any insight into how to coach younger individuals which have this great opportunity to be able to be successful in their careers?
It's a very good question, and first I give you honest answer. Even at the dinner, we had a lot of discussions about other things, not this one. But while I said it's great, there are two things I personally feel that is not so good for young people. And I'll give you one example to start with. So, can I just take a couple of minutes? Yeah. So when I went to Oxford, my head of department, I went there as a fellow, and my head of department, I don't remember when, but maybe in the first couple of weeks, he called me in and in his office, and he said, so what I'm going to work on. It's a 5-year fellowship position, and he's a very well-known physicist. I was in physics then, and I said, Don, I don't really know, but I'm thinking about two problems. And I told him what the two are. And I said, I need a, and this is what he said. And the reason I'm saying is something negative. I think something not so good about young people at the moment. And this is what he said. He said, okay, you come back to me in 3 months and you tell me what you're working on. I reckon now, even I don't know, certainly not in North America, nobody would be a supervisor or supervisor, meaning head of department, who would say, just don't worry about it. Just come back to me in 3 months. Tell me what you're going to work on. They want to know the so-called, hit the ground running idea is too much of it. So I think it's negative for me. I don't think that's the way. Creativity does not work like that. To be creative, you don't hit the ground running. You have to do a lot of preparation. Creativity is about play, so you need freedom. The other thing is another thing that I think we live in a wrong world at the moment is intellectual level, is that negative results is bad result. Negative result is not bad result. It's a very good result, just as good as any positive result. Because negative result is bad for me in the sense I didn't find something. But if we think science, not just for me, but for all of us, then if I can publish negative results, then all of you don't have to go down that cul-de-sac. So it saves ultimately a lot of effort. So this concept that is very difficult to publish negative results, I know, and I think is wrong, but that's the world we live in. So, but I don't want you to be negative about it. So I do think that those two ways are seriously badly, we do it badly. So we as, grant evaluators should think about them. The bigger science, not just individual achievement. That's the problem. Society is built on individual achievement. Individuals, don't get me wrong, does not matter as much as the bigger society. It shouldn't be about Trump. It should be about US. One person is nothing in this world. Now, then coming down, so what can they do? So I think these young people, as you said, so we senior faculty, we should help them. We should help them. So I think it's our job to nurture the young people because they are the future of your department, your country. So it's our job to nurture the young. So we should help them. So that's my advice.